as you mentioned, like there are two major AD risk genes that play outsized role in enhancing the risk for sporadic Alzheimer's disease. That's apolipoprotein E4 and TRIM2 with R47H mutation. And when we combine these two mutations, we specifically look at the immune responses that it enhanced. And the immune response enhanced is very different between male and females. And in the females in particular, the immune responses, including a response that is called type 1 interferon response. And another aspect of the response that is enhanced by these two genetic risks in female immune cells is senescence. So the female microglia carrying these two risk genes become aged faster. So they exhibit senescence phenotypes. And that is very different from the male immune cells carry these two gene risk. So I think that should inform us about the disease progression and mechanism could be different between male and females in the Alzheimer's disease, especially when it regards to the immune response. Mouse are not human, so I think that is something that our researchers have to realize the limitation when we are using mouse models to model disease as complicated as Alzheimer's disease. There are limitations. So to complement the mouse models, a lot of times, using human-derived iPSC cells would give us more confidence with what we have seen in the mouse models will also be replicated in humans. So I think that is one of the directions that we're going is to model the disease in a more human-relevant scenario. So I think that's what I wanted to particularly emphasize is that when the clinical trials are performed, when the biomarkers are collected, the stratification by sex is very, very important. It's very important to know the differences or the of the progression and also the response to the specific drugs in the clinical trials in terms of both the efficacy as well as how the biomarkers change differently between males and females. So that's what I hope our study could bring attention to is the tailored approach from the disease intervention perspective, as well as the tailored approach in terms of disease monitoring and prediction perspective should be stratified by sex.